All right, so now we're going to talk about glucose catabolism. So specifically those steps that we talked about in the end of the previous lecture, glycolysis as well as pyruvate catabolism are going to be all included in this here. Okay, so we talked a little bit about these first few stages where first you go from from having the polysaccharides that you've ingested, converting them to monosaccharides, which make their way into the bloodstream and then are imported into the cell. And then finally, these last steps, which break down the glucose into smaller molecules, which then move into the mitochondria for oxidation. All of these are releasing energy along the way, okay? First is what we're going to talk about is glycolysis. Okay, glycolysis takes place in the cytosol, and it is where we're going to break down glucose from being a 6-carbon molecule into smaller 3-carbon pyruvate molecules. Okay, so essentially this is going to be this process of taking a glucose and breaking it in half into these smaller components. One of the reasons why I was able to combine these multiple chapters into one is because we're going to gloss over a lot of the details of this metabolic step. So I don't expect you guys to have memorized this sequence of converting glucose to glucose 6-phosphate to fructose 6-phosphate, et cetera, et cetera. I don't really care if you know those individual steps. I want you to sort of see the bigger picture of what's going on here. That's the important part. So we don't have to know all of these details. I'm not going to expect you to know all of these details. It's the big picture that we care about. Okay. So glycolysis occurs in a series of steps, right? So on the one hand, we start with glucose and we're going to end with pyruvate. We're going to go through a series of steps. Each one of these steps is catalyzed by a different enzyme. Okay, some of these steps require an input of energy. In fact, the first few steps of glycolysis are what we call the energy investing phase, because here ATP is actually being consumed. So 2 molecules of ATP get consumed in this energy investment phase. And the last one here you can see is we've actually broken up our glucose from being 6 carbons into these 3-carbon components, which will again eventually get converted into pyruvate. The last few steps are what we call the energy generating phases. So you're going to, from these set of steps, you actually create 4 molecules of ATP. So each one of these structures represents 4 ATP. This is ADP going to ATP. So what this figure is showing you, each one of these circles represents a phosphate group. So in these steps, we're actually generating 4 molecules of ATP as well as 2 molecules of NADH. And again, we'll see once we talk about electron transport that NADH will be converted to ATP later. Okay, so both of these, you have this direct output of 4 molecules of ATP, but you also have indirectly an energy output in the form of NADH, which will be converted to ATP later. Okay, so summary. What gets consumed is glucose and 2 molecules of ATP. And what gets generated is 2 molecules of pyruvate, 4 molecules of ATP, and 2 molecules of NADH. So in terms of the net generation, you would get 2 molecules of ATP, right? Because 2 are consumed, but 4 is generated, as well as our pyruvate and NADH. This is not a balanced chemical equation. There's some water that's consumed. There's some protons that are generated. This is a summary of like the meat of what you really need to know from these reactions, okay, from these glycolysis reactions. You should know that it requires 2 ATP, but that 4 ATP are produced, and thus there's a net of 2 ATP. This pyruvate is going to be important. We're going to see it get further catabolized. And then again, we're going to see later on how this NADH gets converted into ATP as well. So the pyruvate. So now that our glucose has been broken into these 2 molecules of pyruvate, that pyruvate is going to be transferred into the mitochondria where it's going to undergo oxidation. Real quick. Let me just this right here is pyruvate. Not that we need to know the structure or anything like that, but it's just kind of good to have this sort of picture in mind, or at least a rough picture of what it is. You see that we have three carbons, and it's also heavily oxidized, right? So there's a carbonyl group and a carboxylic acid group right next to each other. In general, we're going to see this a lot. Oxidation reactions produce energy. So that energy is harnessed in the form of ATP. Okay. All right. So pathways for pyruvate. Again, that glucose is converted to pyruvate in the glycolysis, which happens in the cytosol. That pyruvate, it moves into the mitochondria, where it undergoes further oxidation. There's two different types of oxidation that can occur depending on how much oxygen is present in the cell. The first is under aerobic conditions, meaning oxygen is present. The pyruvate is converted to acetyl-CoA. All right, acetyl-CoA, and carbon dioxide. When oxygen levels are low, the pyruvate is then reduced to lactate. Okay, we're going to see that this has the potential to produce lots of energy, whereas the anaerobic conditions only produces a little. Actually, it requires energy. But under anaerobic conditions, the cell can only generate a little energy, and that energy actually comes from like glycolysis. The reduction of pyruvate actually requires an input of energy. Okay, so, and this is what occurs most of the time, and this is what the cell really wants to do, is carry out these reactions under aerobic conditions. And aerobic means that oxygen, so aerobic means oxygen present. That's what you want to have in your mind. Here we have pyruvate being converted to pyruvate gets converted to acetyl-CoA, and you get an output of NADH as well. Okay, there's CO2 that's output and stuff like that. Again, I'm really focusing on the important components, the ones that are eventually going to lead to ATP, because that's what this is all about. How much ATP do I get from one molecule of glucose? Okay, this acetyl-CoA is going to be important because this is going to go on into the citric acid cycle, which generates even more energy, right? So again, you think back to that summary slide I showed you before. Okay, right, so the whole idea was, with the exception of this last reaction that's specific to lipids, when we're talking about glycolysis here, let me just get rid of all these markings, right? The idea is glycolysis occurs, the products of that go into this pyruvate catabolism, and the products of that into the citric acid cycle, and the products of that into the electron transport chain, right? So all these things are linked. So that acetyl-CoA that's produced is going to become important next lecture. Right, but then again, you also have this payoff of this NADH that's created as well. And again, we'll see that get converted to ATP later. Right, so this is what happens under aerobic conditions. This is what the cell really likes, because this is going to lead to a lot of energy. Under anaerobic conditions, so anaerobic meaning without oxygen, the cell can't do what it wants to do to get rid of the pyruvate, so it has to revert to another reaction, which is known as the conversion to lactate using the enzyme lactate dehydrogenase. And that requires an input of energy in the form of NADH. So we actually lose one of the NADH molecules that we generated in glycolysis if we have to carry out this getting rid of pyruvate under anaerobic conditions. Okay, so if we're under anaerobic conditions, the net of this is now both glycolysis plus the pyruvate catabolism. Here, we only generate the two molecules of ATP that were created in glycolysis. There isn't an additional output of energy in the form of NADH. And in fact, because we had this anaerobic reduction of pyruvate to lactate, it actually consumed the NADH that was generated in the glycolysis step. So the net payoff that you get for having metabolism of glucose carried out under anaerobic conditions is just 2 molecules of ATP. Okay. So just to sort of illustrate here, this is what we discussed under aerobic conditions. Our pyruvate is converted to acetyl-CoA, and we get this extra payoff of a molecule of NADH that's generated. And again, we're going to see acetyl-CoA go on in the citric acid cycle to be created into even more ATP. If we're under aerobic condition, or I'm sorry, if we're under anaerobic conditions, we get the pyruvate converted to lactate, and that actually consumes, the NADH. Okay, so that requires an input of energy. Other organisms have an additional anaerobic reaction that takes place. Yeast, for example, will actually, instead of converting the pyruvate to lactate, it will convert it all the way to ethanol. This is what's known as fermentation. You can see that this also requires an input of energy at this last step. And this is the reaction that we've harnessed making beer and wine, because it generates ethanol, which is, the alcohol that we drink. So this is a reaction that doesn't take place in humans, but is widely seen in other organisms. So again, the benefit of anaerobic conditions is when it's paired with glycolysis, you do generate energy. You do generate 2 molecules of ATP, but we're going to see it's nowhere near as beneficial as carrying this out under aerobic conditions, where you're going to get a lot more ATP at the end. You've generated your 2 molecules of ATP through glycolysis, but we're going to see a lot more energy being generated from this NADH that's created along the way. Okay, so just to summarize, glycolysis produced 2 molecules of ATP. The net result of glycolysis is 2 pyruvate, 2 ATP, and 2 NADH. Under aerobic conditions, that pyruvate is consumed, and we will get acetyl-CoA and NADH. Okay, so let's just make a summary here under aerobic conditions. If we have glycolysis, so let's do glycolysis plus pyruvate, catabolism. Under aerobic conditions, we would get 2 molecules of ATP. We would get 4 NADH, right? And that's because we actually got 2 pyruvates out of it. So we'll get 2 of each of these products. So that would lead to a total of 4 NADH. And we have 2 acetyl-CoA, which we're going to see goes on to produce more energy through the citric acid cycle. Under anaerobic conditions, so again, anaerobic meaning without oxygen, we have our 2 ATP that's produced from glycolysis, but that NADH that we created actually gets used up. And so at the end, all we have is 2 ATP and 2 lactate, and that lactate doesn't actually do anything for us. It doesn't actually lead to more energy like acetyl-CoA does. Okay, so that's why under anaerobic conditions, the real payoff is just the 2 ATP that you get from glycolysis. Under aerobic conditions, we're going to see that we get the 2 ATP from glycolysis, but we're going to see that this leads to a boatload more ATP in further metabolic steps. All right, so let's make sure that we can do this learning check and actually match these two with their description. So what is produced during anaerobic conditions? That would be the lactate. A reaction series that converts glucose to pyruvate, that would be glycolysis. Metabolic reactions that break down large molecules into smaller molecules and energy. Those are what we call catabolic reactions. And substances that remove or add H atoms in oxidation and reduction reactions. We didn't actually talk too much about the coenzymes. The original format of this chapter spent a whole lecture talking about the different coenzymes and their different structures, and I made a kind of decision to gloss over those. The ones that we're going to talk about specifically, NADH, FADH2, and then the coenzyme A that we get to make acetyl-CoA. Those are the big three coenzymes that we're going to talk about. Their main role is in promoting oxidation reduction reactions, but we're going to see that they get converted into ATP energy. Those all fall under this umbrella of coenzymes. But we're not going to worry about their structure or anything like that. So this would be coenzymes. And again, the coenzymes are used to catalyze oxidation reduction reactions, and furthermore, more importantly, really are used to in later metabolic steps to create ATP. So the CoA again goes to acetyl-CoA. All right, so we're going to talk briefly about two anabolic reactions for glucose. We're not really going to go into too much depth with them other than to make sure that we know the terminology. So first of all is the creating of glycogen. Remember that glycogen is the polysaccharide that is found in animal cells. So one of the ways that your cells will store glucose for later use to later be harvested is to synthesize glycogen. Okay, again, glycogen, when you hear glycogen, you think animal polysaccharide. Okay. This occurs in what's called glycogenesis, right? So glycogen is synthesized in glycogenesis. The opposite process, which wouldn't be an anabolic process, it would be a catabolic process, but the breaking down of that glycogen to form glucose, the exact reverse of glycogenesis is glycogenolysis. Okay, so that would be the reverse process. And then the other glucose anabolic process we want to talk about is glyconeogenesis. All right. This is going the exact opposite direction of glycolysis, right? So this is the reverse of glycolysis, where we synthesize glucose from pyruvate. This requires an input of energy. And you might think to yourself, why the heck would the cell go through the process of, bother with synthesizing glucose from pyruvate? Well, there's certain cells in your body that can only use glucose for metabolism. It can't use lipids or anything like that. The brain is kind of an important one. And so in periods of fasting or something like that, where your body doesn't have a lot of free glucose floating around, your liver will actually make a bunch of glucose to be released into the bloodstream so that your brain can absorb it. So again, that's glyconeogenesis, and that's the process of creating glucose from pyruvate. Okay. Let's just sort of before we talk about these individually, let's make sure we got all this terminology that's really, really freaking similar, right? So we have glycolysis, and this is breaking glucose into pyruvate, glycogenesis, glycogenolysis, and then glyconeogenesis. So let's just pick apart these words real quick. So first of all, I want to point out, let me get rid of my markings here, that these two contain the word glycogen in there, right? So this involves glycogenesis is the process of synthesizing glycogen. Glycogenolysis is the process of breaking down glycogen. The word glycogen is found in both of those terms. Okay. The other thing I want to point out, all of the steps that involve breaking stuff down contain the word lysis. right? So lysis is this term for breaking down stuff. In terms of glycolysis, we're breaking down glucose. In terms of glycogenolysis, uh, we are breaking down glycogen. Both of those contain that term lysis, okay? And both of the anabolic terms contain the word genesis. Okay, so genesis is the process of creating something. That's signals that that's an anabolic process, right? Where we're creating something larger from smaller building blocks. In terms of glycogenesis, it's glycogen that we're creating from smaller glucose molecules. And in terms of gluconeogenesis, it's glucose that we're creating from those smaller pyruvate molecules, okay? The other thing, I guess, just to point out, in glyconeogenesis, you have this word neo in there, right? You're synthesizing a new glucose molecule from pyruvate. Okay, so I know that these are all very similar terms that we're going to have to keep separate. So hopefully that kind of helps you parse it out. When you're, when you come across one of these terms, just take a second to pick it apart, pick the word apart, and you'll be able to get it. All right. So taking a look at each one of these, again, to be honest, this is all I expect you to know about these three processes. The fact that glycogenesis is synthesizing glycogen from glucose, glycogenolysis is the exact opposite, going from glycogen to glucose, and glyconeogenesis is synthesizing glucose from pyruvate. That's really what I expect you to know. But just to sort of take a little bit more detailed of a look, right? So in glycogenesis, again, we are generating glycogen, okay? And that is just going from individual glucose molecules through a series of steps that then creates these branched polymers, the glycogen, all right? And this is the way your body can save this glucose for later, for when it really needs it, is to package it into glycogen. There's a series of steps that we don't really need to know the details for, but just like before, there are a bunch of intermediates. Each one of these steps is going to be catalyzed by a different enzyme that's present in the cell. All right, so then glycogenolysis is going the exact opposite direction. So now we're going from glycogen all the way back to glucose. To be perfectly honest, I just lazily recycled this same figure. The individual steps are not the same in terms of they're not catalyzed by the same enzymes. Nonetheless, all I really need you to know is glycolysis is going from glycogen to glucose, whereas glycogenesis is the process of going from glucose to glycogen, right? These are opposite processes of one another. Genesis. Okay. Don't even need to know the individual steps. Okay, and then much like those two are the reverse of one another, gluconeogenesis is the reverse of glycolysis, right? So glycolysis is all about starting with glucose and converting it to pyruvate through a series of steps. Glyconeogenesis is the exact opposite, starting with pyruvate and converting that to glucose through a series of steps. We don't even need to know what those steps are. We just need to know that glyconeogenesis is the reverse of glycolysis. Okay, so this is a little bit of a cheat sheet diagram of all of what we've talked about for glucose here. We have, or really for carbohydrate metabolism, we have the digestion, which is how polysaccharides are broken down into their monosaccharide units. We can convert those to glycogen through glycogenesis, or convert glycogen back to glucose through glycogenolysis. There's something called the pentose phosphate pathway that I am going to just gloss over because I don't think we need to know the details about it. But that's how we create stuff like ribose, which is used in RNA, as well as then later on converted to deoxyribose. So this is how we create those building blocks, those sugar building blocks that we use in RNA and in DNA, which is of course a very important component of the cell. Those are generated through what's called the pentose phosphate pathway. We learned about glycolysis, which is now how we harvest. Now we're starting to talk about how we harvest our glucose for energy. First is through this glycolysis, which we use to convert glucose to pyruvate. If we wanted to go the opposite direction, we would use the glyconeogenesis. And then we talked about how pyruvate gets further catabolized into acetyl-CoA. And this acetyl-CoA will make its way into even more energy through the citric acid cycle, which is the focus of the next lecture. In the absence of oxygen, so this is plus oxygen, that's under those aerobic conditions is how we get our acetyl-CoA. That's what the cell wants to do. If the cell is starved of oxygen, you have this other process, so this is without oxygen, that converts the pyruvate to lactate. Again, we talked about how that's not as beneficial for the cell because it doesn't generate as much energy. And then what can also happen in other organisms is this fermentation where pyruvate is converted to ethanol.